So good afternoon, everyone, and welcome to today's ERN-EuroBloodNet Thursday webinar. We are delighted to have you join us for this session on gene therapy for pyruvate kinase deficiency and congenital dyserythropoietic anemia type 2. Before we begin, just a few housekeeping notes. Your microphone will remain muted during the presentation. Please note that this webinar is being recorded, and you can submit your questions via the Q&A chat box at any time. We'll address them after each presentation. I let now the floor to the moderator, Professor Beatrice Gilbis, ERN-EuroBloodNet co-coordinator.
Okay, so we are very happy to have all our speakers. You see their picture today. So we will speak about gene therapy. Those innovative therapies are very important for all our patients. And all this group is working in a research group called CIEMAT, and this is within a national network in rare disease for in Spain for CIBERER, and then it's also it has also a link with the institution in Madrid. So I give you the floor, and thank you very much for sharing your expertise.
So hello everyone, my name is José Carlos Segovia. I am working at CIEMAT, CIBERER, Instituto de Investigación Sanitaria Fundación Jiménez Díaz, as already Beatriz has mentioned. And I would like first to thank the EuroBloodNet and all of you guys for giving us the opportunity to share some of our results and to give you some inputs in the field of gene therapy for the treatment of hemolytic diseases. So just a first general slide about this, what is gene therapy. Gene therapy is a system in which we can introduce nucleic acid sequences in cells just to allow the expression of the protein, that if this is a protein that is not present or is mutated in the cell, we can restore the function, and then we can talk about therapy and to correct the inherited gene that this cell has mutated. In the hematopoiesis, the target cell for gene therapy is the hematopoietic stem cell, because we know that in this hierarchical structure that the hematopoiesis has, all the mature cells in the blood come from this small population that is in the bone marrow that are the hematopoietic stem cells. Those cells are the only ones in the hematopoietic system that have the capability to self-renew, to maintain themselves for the whole life of the body of the patient. As I mentioned, they have the ability to differentiate in all hematopoietic lineages. So if we address the correction in the hematopoietic stem cell, any lineage coming from this cell would be also corrected. And because of this self-renewal, the hematopoiesis will be long life corrected in the animal or, of course, in the patient. What are the candidate hematopoietic genetic diseases for gene therapy? The major candidates are monogenic diseases, which are diseases that are caused by mutations in only one gene, and this gene is known. Also, the optimal situation is with a recessive inheritance, so in which we need both copies of the gene mutated to generate the disease. That means that if we can correct one of the copies, then the disease would be corrected. And in the case of the hematopoietic genetic diseases, the known of the potential cure of the disease with allogeneic bone marrow transplantation is also a factor that informs us that this disease can be treated by gene therapy. But instead of using allogeneic cells, using the own cells of the patient corrected with the gene therapy tools. We can think nowadays in two different gene therapy strategies. One is based on gene addition in the inclusion of a corrected gene into the cells. And for that, the strategy is called lentiviral gene therapy, which means the use of lentiviral vectors to introduce the corrected copy into the hematopoietic stem cells of the patient. And these vectors are very efficient in introducing genetic DNA in the nucleus of the cells, and are able, they know how to integrate the genetic material into the cell to allow these cells to transmit the correction to all daughter cells, all the major cells. But integration in this case is not controlled, it's a semi-random integration. The gene expression of the healthy gene that we are introducing is driven by an exogenous promoter that is also included in the tool. And of course, because we cannot control the integration, and the endogenous mutated gene is not going to be its expression is not going to be eliminated. But this strategy is very efficient, and it has been already approved. And there are already registered drugs based on this strategy. The other way to do gene therapy is by gene editing. This is a new strategy and less advanced and needs some improvements. This is in general driven by endogenous nucleases that select the place where the DNA material is going to be integrated. So this is a controlled gene-specific integration. We can integrate then the gene into the locus of the mutated one of the gene that we want to correct. And then we can recover the natural expression. We can reduce or even eliminate the expression of the mutated gene. And of course, as I already mentioned, the integration point is controlled. Nevertheless, it still requires some improvements to use these techniques. How does ex vivo gene therapy work? We are talking about, at least up now, in ex vivo strategy. And this ex vivo strategy is represented in this slide, as you can see. So first, the cells are the hematopoietic stem cells, mainly CD34 positive cells, are collected from the patient after its mobilization with specific drugs like G-CSF or Plerixafor. Those cells are collected by apheresis, by blood apheresis, and then the cells are purified, and the progenitor cells expressing the CD34 positive antigen are recovered. And in an ex vivo culture, those cells are exposed to the gene therapy tools that can be either the viral vectors using this lentiviral strategy or the gene editing tools using the CRISPR-Cas based technology. Once the cells are corrected, those cells can be cryopreserved for taking time and to do additional controls, or directly analyzed by some quality controls, and once the cells are good and all the factors are properly studied, these cells are reinfused into the patient. And in the majority of the cases, this patient needs first to be conditioned by chemo, mainly by chemotherapy, to allow the new corrected cells to engraft and also to eliminate the mutated endogenous cells. Up to now, there are a few, as I mentioned already, registered drugs, and I put here some of them and when they were already approved. And I would like to highlight Synteglo, that was approved in 2019, which is an antiviral vector developed for the treatment of sickle cell disease and beta thalassemia. So, in the case of gene editing, there is nowadays one drug already approved a couple of years ago in 2023 for the treatment also of sickle cell disease and beta thalassemia. So what about pyruvate kinase deficiency? Just a few words to talk about pyruvate kinase deficiency. Pyruvate kinase deficiency is a monogenic rare genetic disorder that is characterized by anemia, by a premature destruction of the red blood cells, and it has more than 300 mutations have been already identified in the gene that expresses this protein. The epidemiology of the PKD has a prevalence around 1 in 20,000 people of the Caucasian population, and can say that around 3,000 to 1,000 patients combining US and EU are defined. Although the incidence is not very well unknown, we think that is clearly underestimated. The current treatment options are mainly palliative, based on splenectomy, chronic blood transfusions, with also have remarkable side effects. And the only curative way is the bone marrow transplant, allogeneic bone marrow transplant. So it has all the characteristics interesting for applying gene therapy, and we started to apply this with lentiviral gene therapy strategy. Using this lentiviral vector expressing a corrected version of the pyruvate kinase enzyme, we perform all the preclinical studies in animal models, mainly in the mouse PKD model, and in 2014 and in 2016 we got the orphan drug designation from the European Medical Agency and also from the FDA from the States. With the collaboration of Rocket Pharma, we were able to initiate a phase 1 clinical trial, and 4 patients have been treated, have been transfused up to now. Here is the list of the characteristics of the 4 patients that you can see here. I would like to highlight that in all of them, the average hemoglobin in the 2 first years was around 7, so clearly a very severe anemic patients. And in the 4 patients, 2 adult patients were treated, 1 female and 1 male, and 2 pediatric patients were later on treated as well, also 1 female and 1 male. the number of CD34 positive cells that were obtained after the apheresis, after the mobilization, were good enough to perform the gene therapy, and after the product process and after the culture, a number above 2 million CD34 positive cells per kilo were obtained that were sufficient to perform progenitor's transplant with an efficient vector copy number. That means with an efficient correction in those cells. So after the infusion of the cells, we can see here cells were infused at this point where the star, where you can see the star. This is the history before the infusion, and after the infusion, the blood cell transfusions were completely stopped. The patients didn't require any additional blood transfusion. And if we analyze the level of hemoglobin in the patients, you can see that right after the infusion of the corrected cells at day 0 here, the hemoglobin jumped from around 7 to almost normal levels, around 14 grams per deciliter in the adult cohort, and similarly in the pediatric cohort. Only one patient didn't jump up to 14, but still in the range of 10, the patient is, as we have already seen, transfusion independent. These patients have been already followed, mainly the adult patients, for almost 5 years up to now, and the patients are continue being transfusion and independent and with almost normal hemoglobin levels. So in conclusion of this part, we can say that this RPL301, which is the product, the cells corrected with the gene, is beneficial for the patient, that we have got a sustained clinically meaningful improvement in hemoglobin and in other blood parameters that I haven't shown, and no side effects have been obtained up to now. And just I would like to point out that the phase 2 trial is now under preparation, and probably by the end of this year, 2025, or the beginning of 2026, the phase 2 will be open for PKD patients that would be feasible for inclusion in this trial. So I stop here. I don't know if there is any question or if we can continue. Oh, sorry. And I just would like to thank, of course, the people involved in all this work, and people in the CIEMAT team here. Special thanks to Oscar Quintana and Susana Navarro that developed also this work, and Juan Bueren, the head of the department, that allow us to do this job here. Of course, the people from the Fundación Jiménez Díaz, Hospital Niño Jesús, and Stanford University, all the clinicians and supported people that is taking care of the patients. People are doing the analysis of the cells from the patients, and of course from Germany, from the UCSF, and of course all the people from Rocket Pharma, Elim Nicoletti, Gayatri, Jonathan, Suworth, Kinari Patel, as well as Gaurav Shaw, that believed on us and put their efforts to go ahead with this trial. And now, again, any question, I'm open to answer.
So I can maybe begin.
Yeah, sure. Thank you.
So there are existing treatment like mitapivat and transfusion that address the symptoms, but of course don't resolve all the disease impact. What are your expectations on the disease impact?
Yeah, we know that mitapivat is doing a very good job these days, but we all know also that mitapivat is not working in probably in a number of patients, in a relevant amount of patients that are not responders to mitapivat. So for those patients that are not mitapivat responders and also are not responding to splenectomy, for example, so those patients that do not have any other choice, gene therapy could be the option. But I also would like to add that this therapy is 100% curative. I mean, you don't need to continue taking any kind of drug or any control because it's a treatment that is done once, and then the patient becomes a normal non-anemic patient, which is very, I think that is very important, and is the complete solution. We understand that the protocol is quite complicated and quite harmful because it requires chemotherapy for that. It requires more or less 1 month of hospitality in the hospital to recover. But after that, the patient is completely cured. We are working in the lab in alternatives to simplify this toxic conditioning with alternatives that could be less toxic and easier to apply. But again, compared to other treatments, with this, the patient becomes a normal person.
Okay, I don't see any other questions, so I have another one. Did you soak already at the eligible criteria?
Oh, yeah, at this point, well, I didn't put this slide just for the sake of time. But the eligible criteria are mainly patients that are still transfusion dependent even after splenectomy, and that they do have a history of anemia and blood transfusion requirements in the last 2 years, quite important, 2, 3, or 4 requirements.
Thank you very much. And I saw one question in the chat. Where is the trial going on?
The first the phase 1 trial is already closed, as I mentioned, and the phase 2 trial, when open, it will be run in Europe and in the States at the same time. And for now, hospitals in Madrid, Spain, and hospitals in California, in Stanford, are the hospitals that will take care of the trial. But I know that at least one additional hospital will be open in Europe and another one in the States to be able to assist the number of patients needed.
Yeah, so the last question, because otherwise we will be over time. So what is the cost involved if someone wants to participate in trial?
the trial is sponsored by Rocket Pharma, and if the patient is eligible for the trial is recruited, the cost will be covered by the company, by the pharma company that is sponsorizing this trial.
Thank you very much. So let's go on. And you can always put any question in the chat, and we can take them after the presentation. Don't be shy.
Okay, so shall we start? So good afternoon, everyone. I'm Isabel Ojeda-Pérez. I'm a postdoctoral research at José Carlos Segovia's group. And today I will talk briefly about what we are doing in gene editing for pyruvate kinase deficiency. So as José Carlos already mentioned, lentiviral gene therapy is characterized by a semi-random integration of the transgene that is driven by the exogenous promoter expression. So since the integration of the transgene is semi-random, it does not alter the abnormal expression of the mutated gene. For that reason, an ideal gene therapy will involve introducing the transgene into a controlled manner, driving this expression by the endogenous promoter of the gene, and also disrupting the expression of the mutated gene. In this context, gene editing emerged as a potential tool to precisely correct mutation in different cell types, such as hematopoietic stem cells. So the most targeted nuclease used worldwide nowadays is CRISPR-Cas9 system. So with CRISPR-Cas9 system, what we are doing is to introduce double-strand break into the DNA. Those double-strand break can be repaired by two different pathways: non-homologous or microhomology end joining, or homologous directed repair. Since we are focused on correcting most of the mutations that are causing pyruvate kinase deficiency, we focus on homologous directed repair pathway. So this HDR is just occur in S and G2 phases of the cell cycle, and it needs a donor template to repair this double-strand break. So what we did in the lab was to design a donor template that were able to introduce our corrected version of the gene, the cont-optimized version of the RPK gene, just next to the regulatory region of the gene. So in this case, we are using the endogenous promoter of the gene. So to introduce all of these gene therapy strategy, what we used was electroporation. With electroporation, what we are doing is doing small holes in the membrane of the cells that allows the ribonucleoprotein enter inside the cell. So we combine this electroporation and the ribonucleoprotein system with an AAV6 donor that carry the donor template that I've already told you that we are using to correct the disease. So the strategy that we are proposing here, it's a non-gene genome editing strategy that combines both CRISPR-Cas9 system and AAV6 as a vector to deliver our donor template. So what we did in the lab was first to purify the hematopoietic stem and progenitor cells, either from cord blood or mobilized peripheral blood. We took those cells and pre-stimulate these cells during 48 hours. After this pre-stimulation time, we electroporate the cells and transduce the cells with the AAV6 vector that carry our donor template. 24 hours after, we wash the cells and study the gene editing of these cells. And also we took the cells and transplanted into immunodeficient mice. So first, what we studied was the amount of virus that we need to obtain the highest levels of gene editing with the less toxicity in these hematopoietic stem and progenitor cells. What we observed was that using this MOI, that was MOI 2000, we obtained at least 60% of gene editing in vitro with no toxicity. So what we did was to took these cells and transplanted into immunodeficient mice. First, we studied the level of engraftment in those mice 2 months after transplant, and we observed engraftment in bone marrow, spleen, and peripheral blood of those mice. We also studied the levels of gene editing in those mice in the different lineages that we sorted, that were CD44, CD45, CD34, CD33, and CD19 population. In all of the cases, we were able to see similar levels of gene editing. So once that we optimize our gene editing protocol in wild-type hematopoietic stem cells, we wanted to apply this protocol into PKD samples. So as we did before, we took PKD samples, purified the cells, and edited with our ribonucleoprotein and AAV6 donor. As José Carlos mentioned, these PKD cells have an impair in the production of ATP. So to study if we were able to restore the phenotypic function of these cells, what we did was to differentiate these cells into erythroid lineage and study the levels of ATP in those edited cells. So what we observed was summarizing this graph. As you can see here, the white bars are the levels of ATP in healthy donors, and in the light gray bars are the levels of ATP in the edited cells. So as you can see, we treated 4 different PKD patients, and in all of 4 treated patients, we were able to restore the levels of ATP compared to a healthy donor. As we did before, we also transplanted those cells into immunodeficient mice. Those cells were able to engraft. We obtained a good percentage of gene editing in those cells. And again, we measured the levels of ATP comparing unedited and edited cells. And as you can see here, we were able to restore the levels of ATP also in transplanted mice. So just as a map, we developed in the lab a knock-in system that combined ribonucleoprotein and AAV6 as a donor. And we obtained high efficiency of gene editing, targeted human hematopoietic stem cells with no off-target effect. Also, when we transferred this information into PKD samples, we were able to restore the energy defect in vitro and in vivo. So here we develop a gene editing strategy that is a feasible strategy to apply for future clinical application to treat PKD patients. And now we are going to move to another anemia that is CDA2, and it's going to be presented by our colleague Mercedes Dessy Rodríguez.
Thank you, Isabel, for the introduction. Good afternoon to everyone. Now we are going to move to another disease that we are working also here in CIEMAT, that is congenital Dyserythropoietic anemia type 2 or CDA2. And I'm going to show you the result of the gene therapy that we have developed for this disease. First of all, a brief introduction about this disease. CDA2 is a rare inherited disorder with a very low incidence, around 0.71 cases per million. Patients with this disease are characterized for present ineffective erythropoiesis, and one of the main characteristics of this disease is the accumulation of binucleated erythroblasts in the bone marrow. This disease is caused by mutation in the SEC23B gene. Main of the clinical signs are anemia of varying degrees, often accompanied by jaundice, splenomegaly, and iron overload. Nowadays, most of the treatments are palliative, such as periodic transfusion. 20% of the patients are transfusion dependent, iron chelation, and splenectomy that is only recommended in several cases. The only curative treatment described is the allogenic bone marrow transplant, which implies additional limitations, such as the availability of HLE matched donor and the possibility of the graft versus host disease. Therefore, it is necessary to look for new therapeutic strategies to cure this disease. However, one of the main limitations that we find in this development is the limited access to CDA2 patient sample to work with due to the low incidence of this disease, together with the absence of an animal model able to reproduce the CDA2 human phenotype. It is because the SEC23 mouse deficient for the SEC23B deficient mouse show pancreatic alteration instead of a red blood cell phenotype. Therefore, our main objective with this project is to develop new therapeutic approaches for CD8. And for that, we propose two different objectives. The first one is the development of a human cellular model able to reproduce the main characteristic of CD8 patients, and which we are going with in the development of new therapeutic strategies. And the second objective is the development of an antiviral-mediated hematopoietic stem cell gene therapy. As José Carlos told you very briefly, our objective is to collect hematopoietic stem cells from CD8 patient, correct them with the therapeutic vector, and then reinfuse again into the patient these corrected cells. Now we are going to focus on the first objective with the development of a CD8 cellular model able to reproduce the main characteristic of CD8 patient. For that, we purified hematopoietic stem and progenitor cells present in cord blood, and we nucleoaffected these cells with the CRISPR-Cas9 system to disrupt the expression of the SEC23B gene, generating then SEC23B knockout cells. First of all, we measured the SEC23 protein expression level in these knockout cells, and we observed a clear reduction in the SEC23B protein expression in comparison with the control condition. Next, we also wanted to evaluate the phenotype of these knockout cells during the erythroid differentiation process. For that, we developed here in the laboratory a 14-day protocol in which we add different cytokines to a basal medium to differentiate the hematopoietic stem and progenitor cells to major erythrocytes. At the end of this erythroid differentiation protocol, we measured the erythroid phenotype by flow cytometry, and as we can see here in this graph, the number of most major erythroid cells that here are represented in the red is diminished in the knockout cells with respect to the control condition. Moreover, we analyzed the erythroid phenotype also by microscopy, and as we can see here, we observe a significant increase in the percentage of binucleated erythroblasts in our knockout condition with respect to the control cells. So with all this data, we can say that with our CD8 cellular model, we are reproducing some of the main characteristics described for CD8 patients, such as a reduction in the number of the major erythroid cells, together with an increase in the percentage of binucleated erythroblasts. Once we generated our CD8 model, we moved to the second objective, that is the development of a lentiviral gene therapy for this disease. For that, we designed a lentiviral vector carrying the wild-type version of the SEC23B gene and under the control of the ubiquitous promoter PBK. Taking advantage of our SEC23B knockout cells, we proved the efficacy of this therapeutic lentiviral vector. And for that, we transduced the knockout cells with different multiplicities of infection of the lentiviral vector and performed an in vitro erythroid differentiation. First of all, when we measure by Western blot the SEC23B protein expression, we observe a slight increase in this protein expression level in all of the transduced conditions with respect to the untransduced knockout ones. And more interestingly, when we analyze the erythroid phenotype by flow cytometry at the final point of the erythroid differentiation, we observe that in the transduced condition, there is a higher number of major erythroid cells in compared to the untransduced knockout cells. And moreover, when we analyze the percentage of binucleated erythroblasts, we observe a reduction in the percentage of binucleated cells in our transduced condition with respect to the untransduced one. Therefore, with our therapeutic lentiviral vector, we are able to correct the main characteristic of CD8 phenotype, at least using our CD8 cellular model. Next, we have the opportunity to receive some peripheral blood from CD8 patient in the laboratory, and we also proved the therapeutic efficacy of the therapeutic lentiviral vector in this sample. For that, we purified the CD34 positive cell present in this peripheral blood, and we transfused them with the lentiviral vector. Here, I want to remark that the number of CD34 positive cells that we can purify from the peripheral blood is really limited, around 2,000 CD34 positive cells. But despite this low number of cells, we were able to transfuse them and also to perform an in vitro erythroid differentiation. When we measure the SEC23 protein expression level by Western blot, we observe a slight increase in the patient in the cell of the patient transfused with the lentiviral vector with respect to the untransfused cells. And more interestingly, when we analyze the erythroid phenotype by flow cytometry, we observe a higher number of mature erythroid cells in the transfused condition with respect to untransfused one. And also the phenotype of this transfused condition was more similar to the healthy donor sample. And finally, when we analyze the percentage of binucleated erythroid cells in 2 of the transfused CD2 patients, this percentage of binucleated erythroblasts was lower. However, none of these results showed significant differences. Finally, to sum up, I want to tell you that we have generated a CD8 cellular model using the CRISPR-Cas9 system, and this model is able to reproduce the main characteristic of the CD8 phenotype. And using this model and also a CD8 patient sample, we have developed an antiviral gene therapy that partially compensated for the SEC23B deficiency. And finally, I also want to introduce you the survey we have developed here at CIEMAT, which is aimed at PKD and CD8 patients, and also to their families and friends. With this survey, we want to assess the quality of life of this patient, and more interestingly, what is the knowledge of this patient about the available treatment, especially if they know about gene therapy, and what are their opinions about this. Also to finish, in September, the 15th of September, we are going to have another webinar. In this case, it will be dedicated for patients. So we will be very grateful if you share this information with the patient, and also if you show them the survey and they can complete it. Thank you so much for your attention. And well, now, finally, I would like to thanks to all the people working here at CIEMAT, to all of our collaborators, the clinicians, the patient and healthy donors, also to the EuroBloodNet webinar organizer for giving us the opportunity to present our work here today, and of course, all of you for attending this webinar. And now it's time for questions.
Thank you very much. So I don't see any question in the chat. So did you already contact clinicians for discussing the gene therapy in CDA type 2?
Yeah, we have just contact some clinicians, especially here in Spain. We are also in close contact with Paola from Italy. So thanks also to the clinician, we are receiving some peripheral blood sample here in the laboratory. But for us, it's really important this as a close contact with clinicians, also to receive the sample and to inform them about the evolution of this project.
Thank you. Of course, there is no until today, there is no really targeted therapy for CDA type 2. And we only treat the symptoms, and that's it, of course. So that might be a place for it. So maybe a question. So one question is if cord blood cells required for CDA type 2 patients?
No, cord blood cell is required because for us it's the easiest way to obtain hematopoietic stem and progenitor cells. For us, it will be more useful if daily we can use a patient sample, but it is not possible due to the low incidence of the disease. And also because, for example, bone marrow aspiration is not a usual technique with these patients. So we only can obtain peripheral blood sample time to time. The reason for using cord blood cell is because it is more easy for us to obtain.
Okay, thank you very much. I don't know, I don't see any more question in the chat. So, yes, there is still one. So some of the person in the audience is really active. Thank you very much. So, if the donor is sibling, then both samples are required.
What is the question? Sorry.
If the donor is sibling, both samples are required.
Maybe I can. Hello,
Yeah.
this is José Carlos. In the beauty of the gene therapy is that we don't need a donor. I mean, if you have a sibling, this is gonna be allogeneic bone marrow treatment, allogeneic bone marrow transplant. That, of course, if this is a sibling, the potential graft versus host disease and other diseases associated to the transplant should be addressed. The beauty of the gene therapy is that we are using the cells from the same patient that is going to be treated. So they are their own cells, and there is not going to be any kind of immune reaction or rejection or whatever. So with these gene therapy strategies, either by lentiviral vectors or by gene editing strategies, we completely avoid the side effects and the adverse effects related to the reaction of the patients and the donor cells.
Thank you very much. So there are no more questions. So thank you very much. And of course, the webinars will be available on our website. And I don't know if Christophe, you want to tell something else.
Yeah, I would just like to recall to the survey that we will send by mail. So always survey after the webinar, and don't forget to not to miss the next webinar that we have next Thursday. You have the link to register on the chat of this Zoom. Thank you all for the coming, and thank you for Beatrice Gulbis, the moderator, and the CIEMAT CIBERER team for their sharing the insight they shared today. It was very, very interesting. Thank you.
Okay, it's not the end. So there is still a question.
Sorry.
When do you think that we should have trial clinic on CDA?
Oh, yeah, difficult question.
I knew.
Question. Yeah, we have now the proof of concept. Well, maybe. Well, I will answer this.
Okay.
We have now the proof of concept that it works. And there is still a long way we need to develop, and the preclinical data, some safety and biosafety data, and then apply for probably for orphan drug designation. And with all this work, we could address some companies to ask them to help us to go for the clinics with the CDA2. A few years yet, unfortunately, but we will do our best.
Yeah, I'm sure.
Address as soon as possible.
Okay. No, I really think it's the end. So
Okay.
I wish you a very nice evening. I hope it's not too hot in Spain still.
It is, but anyway, we can.
Okay. Because in Belgium we lost 20 degrees.
Oh yeah, no, we are still in the 40s. So
okay, have a very nice evening, and thank you very much for sharing this expertise really needed for our patients.
Okay, thank you very much.
Bye.
Also, I would like to pass your thanks for my colleagues Isabel, Oscar, and Mercedes, that they are here. So thank you.